All right, we're going to get started. Good afternoon. Welcome to the Department of Pharmacology seminar series. It's my pleasure to introduce Mike Emauele. Mike is an associate professor in the Department of Pharmacology at the University of North Carolina, Chapel Hill. So I met Mike during the COVID pandemic. We just sort of connected based on our common interest in ubiquitin regulation of signaling. Of course, our focus is on GPCRs, and he's looking at ubiquitin regulation of signaling in cell cycle and cancer cell proliferation. I gave a Zoom seminar in the Department of Pharmacology at UNC Chapel Hill, I believe almost 2 years ago, and had the pleasure of meeting Mike. And then we connected to talk a little bit more about Deubiquitinases, which is part of his talk today. Mike received his undergraduate degree at Bucknell University. If Tracy Handel is on the call, yay, yay, Bucknell. And then he received his Ph.D. at the University of Virginia. He then went on to Harvard Medical School, where he worked with Steve Elledge, and there, I think, really developed his interest in ubiquitin regulation of cell signaling. Mike has done incredibly well as a professor at UNC Chapel Hill, has multiple NIH grants, and is also a passionate and dedicated mentor. He received the Excellence in Mentoring Award, which was an award that is student nominated from the UNC graduate program, and has been very much involved in a variety of different initiatives and activities at the university, creating more equitable and inclusive environments. So with no further ado, I'm going to let Mike give his talks.
All right, so thank you for the introduction, JoAnn. Yeah, so I went to JoAnn's talk on Zoom, and I remember this, and I thought I was going to a G protein talk, which it was, but then it had all this stuff that, sorry, I'll try not to talk with my hands too much and hit the microphone again. It turned out it was all about ubiquitin signaling and G protein pathways. So I was super stoked about that because we work on the ubiquitin pathway ourselves. So I reached out and then we ended up having a really nice chat on Zoom one day. And now here I am in San Diego visiting. It's beautiful to be in San Diego because it's beautiful in San Diego. It's good to meet JoAnn in person. I've made some new friends already this morning. I'm going to get to also see some old friends while I'm out here. So thank you for hosting me. And I guess without further ado, I'll get started. Okay, so before I talk about any science, in the chance that I run out of time at the end, I'll quickly thank my lab. Let me get, I was told that I should use the laser pointer here. Okay, so this is my lab. Basically, all of the work I'm going to tell you about today was done by, pointer lags a little bit. This woman in the front with the red sweater on, Taylor, who's actually last day in the lab is Friday, sadly. She's an amazing graduate student. The second project I'll tell you about is from this fantastic postdoc in the lab, Thomas. But as you can see, we have a pretty small group. Most projects are sort of all hands-on-deck affairs. So thank you to everybody here. Okay, so we are interested in cell cycle control. I think probably everybody in the room and also in the room at home on Zoom is familiar with the cell cycle. It's the process by which one cell becomes two cells through the copying of all of the genetic information, and then its segregation into two daughter cells during mitosis or during cell division. I'm not going to throw a whole bunch of cell cycle names and pathways up at you that you don't need to remember. What I want to tell you on this slide, though, is that progression through the cell cycle is driven by changes in the expression of genes and proteins. So this is sort of epitomized by the cyclin-dependent kinases, which I'm sure you probably all heard of. These are kinases that drive a whole variety of events in the cell cycle, and their temporal dynamics and activity are driven by the expression of these proteins called cyclins. They're binding partners. I'm showing you three of them here, cyclin E, A, and B. These proteins go up and down as cells go through the cell cycle. They bind to different cyclin-dependent kinases to control their activity. But it's not just the cyclins that change in the cell cycle. In fact, there are many, many, many hundreds of proteins and genes that change in both expression and protein abundance during the cell cycle. So on the left side of my screen, you can see this is an old now mRNA expression experiment that was done in HeLa cells like 20 years ago. And what you're looking at is the hundreds of genes that are changing during each cell cycle. And on the right, this is a proteomics study that we contributed to with a great collaborator named Tony Lee when he was in Angus Lamont's lab. And what you're looking at again is a heat map of proteins changing in the cell cycle. There's a lot of overlap between the genes and the proteins that change in the cell cycle. And as I'm sure you can appreciate, this is controlled by a whole variety of processes that are changes in transcription and translation that lead to the accumulation of genes and proteins. And then what I'm going to talk about today is the abrupt change in their dynamics as they go away, which is controlled by protein degradation. So this is a very quick overview of the major protein degradation system in human cells. It's called the ubiquitin system. It's a series of enzymes called E1, E2, and E3. What you really have to worry about today are these E3 enzymes. These are the enzymes that really drive protein degradation by specifying substrates for ubiquitination. So the addition of ubiquitin, itself a small protein onto substrates, can then form these long polyubiquitin chains through multiple rounds of ubiquitination. And then these substrates are carried to a big complex called the proteasome, where they're chewed up and destroyed. And there's probably upwards of 600 ubiquitin ligases in human cells, where there's only probably about 35 E2s and a couple of E1s. And really what that tells you is that the E3s are where the specificity in the system lies. They're choosing who's getting ubiquitinated and when that ubiquitination is happening and what that ubiquitination looks like. So within the cell cycle, we know that there are many proteins that are ubiquitinated. I'm putting this up just to show you that there's a lot of cell cycle ubiquitinated proteins, not because I want you to remember any one of these in particular, but to highlight the importance of this process. And if maybe your thing isn't the cell cycle, but you're interested in disease, I also want you to know that there are many oncoproteins and tumor suppressors that are ubiquitinated. So this is a hugely important signaling pathway, both in normal homeostasis and disease. So you can see up here, the MYC oncogene, cyclin D, also cyclin E, P53, NRF2, HIF, the cyclin kinase inhibitors. These are all primarily controlled through ubiquitination. In fact, if you look at P53 as a prime example, its levels at least are not really controlled transcriptionally at all. It's almost controlled completely to the level of to the degree of degradation mediated by its binding partner MDM2. So the key challenge, at least one of the ones that I've been thinking about in this space for a really long time now, is how do we connect enzymes with their substrates? Or kind of in reverse, if we have a substrate that we know is ubiquitinated, are there ways to map it back onto a specific enzyme? We think this is akin to mapping sort of any canonical signaling pathway like the one I'm showing you here, which we know is really important in disease, the RAS, RAF, MEK, ERK pathway. And this is a real challenge in the ubiquitin pathway, as it is for kinases as well. And it's a challenge for a whole bunch of different reasons. So first, the interaction between enzymes and substrates is incredibly fleeting. So these things fall apart really quickly. There's a problem of stoichiometry, and that is that one E3 can often exist in a large protein complex, and that complex can often control many, many dozens of different substrates. So if you, for example, pull on the E3 ubiquitin ligase, for every molecule of that E3 you pull out, you're going to pull out maybe one of, say, 20 or 30 different substrates. By virtue of the fact that these substrates are turned over all the time, many of them are low abundance. And in fact, the binding between enzymes and substrates is really tightly regulated. So maybe it depends on a particular post-translational modification in the enzyme or the substrate. In our case, maybe the cells need to be in just the right place in the cell cycle for that binding to occur. But when that binding occurs, then the substrates all go away because they get degraded. So this makes trapping these interactions really tricky. And we do this all the time. We put tags on different proteins to follow them in space and time or to pull them out. And sometimes these tags interfere with binding. So overcoming these sort of vast challenges is something that we're super interested in. I wish I could tell you that we figured out the trick to find it for all the different enzymes in the ubiquitin pathway. I unfortunately think the answer is that different enzymes and different substrates are going to require different methods. And I'm going to tell you about two approaches that we've taken in the last several years to sort of circumvent some of these challenges. So sort of the overarching goal of the lab is to define enzymes, substrates, and mechanisms of ubiquitin signaling in normal cell proliferation and homeostasis, and also when we can, how it impacts cancer cell proliferation. The two studies I'm going to tell you about today The first are shown in an overview here. The first one involves using systems genetics that allowed us to identify an enzyme substrate relationship that we think is really important in normal proliferation and probably cancer proliferation as well. And what I mean by systems genetics will become more clear in a moment. And in the second one, we use cell cycle transcriptional dynamics together with existing protein-protein interaction networks to identify a new deubiquitinase involved in cell cycle. And I'll tell you about that. The one theme that sort of holds these two things together, besides them all being focused on cell cycle and ubiquitin, is that they leverage large existing data sets that are in the public domain. I think this is something that many of us often overlook. That, I don't know, when I was in graduate school, there were no like giant repositories of protein interactions or transcriptomes of thousands of tumors, or even loss of function genetic screens, which have now been done by the Broad Institute in over 1,000 cell lines. And I think that there's a lot that we can learn from this stuff to generate hypotheses. So the first project that I'm going to tell you about was recently published. I'm not going to go through it quickly, but I'm not going to show you every piece of data from it. But I really want to highlight sort of the methodology we use to get into this project. And this is a systems genetics analysis we did to identify cell cycle E3s in a particular substrate. So this project starts with an analysis of something called the DEP map, or the dependency map. This is an initiative driven by the Broad Institute with other partners, aimed at identifying essential genes or specific cancer vulnerabilities. And the data set, when you look at it more holistically, allows for what we call systems genetics analysis. So these are loss of function CRISPR screens that have been done targeting approximately 18,000 genes. And these screens have now been done in approximately 1,000 cell lines. Last I checked, it was something like 1,100 cell lines. What they do is they make lentiviral CRISPR libraries, they infect cells. They then just simply passage these cells for a couple of weeks. They then recover all of the guides from those cells, either before or after starting the experiment. And they look to see what the representation of those guides are at the beginning and the end. So if you come over here, so this first, in this cartoon, this burgundy bar would represent some gene that really has no effect on cell fitness. It was in equal representation at the beginning and the end of the experiment, whereas this orange one down here, this was some essential gene that really killed all the cells in which it infected because it knocked out that gene. Whereas maybe this green one, this looks more like a tumor suppressor. The cells are actually growing a little better than would be expected when you knock out that gene, right? So again, there's this enormous data set, 18,000 genes, more than a cell lines, all publicly available. There's a really nice website you can go and browse called the DEP map. I don't work for them, but it's a really amazing resource. You should check it out. So if you download the data and you start to play with it, you see the data looks something like this. So this is, each dot on this graph is a different cell line. This is looking at the retinoblastoma gene. This is a tumor suppressor. You can see what you knock it out, cells grow a little bit better than normal. So on the y-axis here, I have the fitness score. That's basically a measure of the representation of a whole bunch of CRISPR guides to that gene versus the beginning and end of the experiment. You can see the cells are growing a little better than normal because you've knocked out a major tumor suppressor. If you look by contrast at the knockout of cyclin-dependent kinase 1, this is the essential mitotic kinase required for cell division. There's really no way of getting around not having this. And you can see sort of universally, all of the cells are super sensitive to the loss of CDK1. The other thing you can do is you can compare those different fitness scores. So this starts to get into this systems analysis I'm talking about. So if you compare the fitness scores for CDK1 versus RB, there's really no correlation between them, right? Because they're involved in totally different biological processes. You can see again, the CDK1 fitness score is shifted way to the left because it's killing all the cells when you lose CDK1, whereas RB is above the axis. But there's no correlation between these things. This is in contrast to looking at the scores for cyclin E and CDK2. These are the kinases and its binding partner that drives S-phase progression. Actually, these are pretty well correlated. And that's because they're in a complex together and they function in the same pathway to drive proliferation. So there are some cells that care more or less about losing CDK2 and cyclin E. But if you care about losing one, you really, really, really care about losing the other. And that's because genes and functionally related either pathways or protein complexes are going to similarly affect fitness across all of those cell lines. So you may have some cell lines that don't care about losing a gene and some cell lines that do. But if you care about losing one gene, say in a pathway or one protein in a pathway, you're going to care about the entire pathway. And the same for a complex. And this is really nicely illustrated by that pathway I talked about earlier, the MAP kinase pathway, where if you say what genes are similar to, say, MEK, which is the center of the pathway, you can calculate the Pearson correlation for MEK versus all 18,000 other genes across 1,000 cell lines where this has been performed. And if you do that, you see MEK is most highly correlated with itself. In fact, it's perfectly correlated with itself. Whereas the top two hits are RAF and ERK. So this is like really amazing. This works really well for basically anything that affects either cell fitness or growth or proliferation. This works really well for autophagy. I don't know anything about the autophagy pathway, but I do know that all the autophagy genes start with the letters ATG. And you can see like all of the top hits for ATG7, which is a component of the autophagy pathway, or all these other autophagy genes. This works for the TNF, NF-kappa B pathway. Again, this is a pathway I'm only partially familiar with, but I know the names of the proteins involved. And also for the cell cycle aficionados out there like myself, you can see we've captured almost the whole G1S regulon here by doing this simple analysis. We were interested in applying this sort of analysis to the Cyclin D CDK4/6 pathway. So this is the kinase, the CDK that's important for G1 phase progression. So CDK4 and CDK6 pair with Cyclin D. They then phosphorylate the retinoblastoma tumor suppressor. And this drives the expression of a whole bunch of genes that are important in S phase progression. We know this pathway is really, really important in both the etiology and treatment of cancer. So many tumors lose retinoblastoma function. This uncouples CDK activity from E2F and allows tumors to proliferate without control. In the last several years, the FDA has also approved a suite of drugs targeting CDK4/6. These are called palbociclib, ribociclib, and abemaciclib. These inactivate CDK4/6 and sort of reawaken the tumor suppressor activity of RB. And these drugs are used commonly now to treat women with hormone receptor positive metastatic breast cancer. And while these drugs have been really successful at extending progression-free survival, it's also clear that there's a lot of challenges to their use. So they've been approved for 7 years now in breast cancer, but have not been approved in any other tumors. The mechanism except mantle cell lymphoma, I should say, sorry. The mechanisms of resistance are, I think, like a total mystery. So essentially no patients derive resistance from mutations in CDK4/6 itself. So there's no kinase mutations there. They never lose RB. I always say if I were to ask this on an exam, like how do you get resistance in this pathway? The students should probably tell me you lose the tumor suppressor. That happens in maybe 5 to 10% of patients. So for the other 90% of patients that might get these drugs, we have no idea what the mechanism of resistance is. And this is a huge patient population. Remember, 300,000 women are going to be diagnosed with breast cancer in this country this year alone. 200,000 of them are going to have hormone receptor positive disease. And we also have no biomarkers for which women should get this drug and which won't, even though we know a quarter of them will not respond at all. So we applied this systems genetics analysis to learn a little bit more about this pathway. And you can see the gene most correlated with CDK4 was this gene Cyclin D, which is what binds it and activates it. So that made us feel like, well, at least this analysis is going to work for this pathway. The 9th most highly correlated gene is a gene called Cyclin F. And we're going to spend some time talking about Cyclin F because we were excited about this because this is one of our favorite ubiquitin ligases, it turns out. When we redid this analysis and centered it on Cyclin F, we could then see that the RB-like protein RBL1, CDK4, Cyclin D, and RBL2, these were all in like the top 100 most highly correlated genes. And that suggested to us that Cyclin F, which I'll tell you a little bit more about in a second here, was doing something really important in this pathway that we had not yet appreciated, and we wanted to figure out what that might be. So what is Cyclin F? So it sounds like it's a cyclin, but it's not a real cyclin, I would say. It oscillates during the cell cycle, but rather than binding to a cyclin-dependent kinase, it's actually a substrate recruitment module for these SCF family ubiquitin ligases. These ubiquitin ligases have a Col1 backbone. They bind to this adapter protein here. And there's about 70 of these FBOX proteins in human cells. Cyclin F is one of those substrate recruitment modules. Virtually all of its substrates, some of which my lab has described, others have been described by both the Pagano lab and the Giangiolella lab and others. Almost all of its substrates are involved in cell cycle control. Cyclin F is itself cell cycle regulated. And it's, as I said, it's a non-canonical cyclin. I don't want you to get confused. It does drive cell cycle progression, but it does so not by binding to a CDK and promoting phosphorylation, but instead by driving ubiquitination. So we constructed 2 cell lines where we could drive cyclin F expression using doxycycline. And we checked basically all of the genes in the CDKRB pathway, which based on our systems genetics analysis were related to cyclin F. And you can see we checked P130, P107. These are RB-like proteins, cyclin D, CDK4, CDK6, and then some controls at the bottom. And the only protein that reproducibly changed was this protein called P130. It's also called RBL2 for RB-like protein 2. So we sort of draw the RB pathway sort of the way I've drawn it in the top left here. And you see this in textbooks all the time, but it turns out it's actually much more complicated than that. In addition to RB, there's 2 RB-related proteins, RBL1 and RBL2, also called P107 and P130. Whereas RB sits on the transcription factors that drive cell cycle progression. These other 2 form something called the DREAM complex, which sits on the same cell cycle promoters to repress proliferation, also by locking down cell cycle gene expression, just in a slightly different way. We could show that if we knocked out cyclin F, so we took away the ubiquitin ligase, P130 levels go up. This is done by CRISPR. We could similarly do it by siRNA. We also show that when we overexpress cyclin F, you can see here, P130 levels go down. And we know this is dependent on the ubiquitin pathway because we can rescue it with MG132. This is a proteasome inhibitor. We can also rescue it by treating with netylation inhibitor called MLN4924. And this is used to inactivate the specific type of ubiquitin ligases that cyclin F assembles into. And finally, we know that this is specific for cyclin F because when we overexpress a whole bunch of other FBOX proteins that have been implicated in cell cycle control, you can see none of them knock down P130 levels, but overexpressing this one ubiquitin ligase completely destroys it. We also had performed a whole proteome analysis using mass spectrometry with tandem mass tagging, where we identified upwards of 7,000 proteins. And we actually captured, this was sort of done retrospectively after the case, but we found P130 or RBL2 among the most statistically significant downregulated proteins in that analysis. So further confirmation that P130 is likely a cyclin F substrate. We took the protein apart. This is like one of these cartoon slides that summarizes like a year of a student's life. She broke down P130 into a bunch of different fragments. She found that the minimal domain that could be regulated by cyclin F was this flexible spacer domain. There were 2 putative cyclin F binding sites in there, which I'm showing you here. It turned out it was this second one that was required for degradation. I'm going to show you a little bit of that data here. So here you can see if we overexpress cyclin F, P130 gets degraded. We mutate that first site, still gets degraded. We mutate that second site, that RRL, to ARA, sorry. The double alanine mutation, I'll just call this P130AA. You can see we totally block degradation. We did an in vitro binding assay where we purified these proteins, and you can see that that AA mutant also no longer binds to cyclin F in the test tube. Together with Seth Rubin's lab at the University of California at Santa Cruz, we established an in vitro binding assay where we used purified cyclin F together with a fluorescently labeled P130 peptide. You can see they bind nicely together with a KD of about 0.2 micromolar. And we could compete them off of each other using wild type P130, that's this blue line here. But if we used an AA peptide that doesn't bind very well, that corresponds to that AA mutant I just mentioned, you can see the binding, the competitive binding was about 50 times worse. We could also reconstitute this ubiquitination completely in vitro. This is done in collaboration with Nick Brown's lab at the University of North Carolina. He's a spectacular enzymologist and biochemist. You can see here, these banding pattern is indicative of P130 getting ubiquitinated. Again, all of these components had been purified from baculovirus or from bacteria. if we use that AA mutant P130, we get essentially no detectable ubiquitination. So this is also the first time anybody's ever reconstituted the SCF Cyclin F complex. We think this is actually really important because it sort of avoids the concern that what we're looking at in cells is perhaps indirect. So then that sort of raised the question, well, why is this actually important, right? So is this degradation? We know that the phosphorylation of, say, P130 and the other RB proteins is important. That's been well established. But what about the degradation? This had never really been tested because nobody had alleles of an RB-like protein that could really allow you to ask this question. So we designed cell lines or engineered cell lines where we could either express P130 wild type or that mutant under control of doxycycline. When we express them, you can see that the AA mutant accumulates to higher levels because it's not getting degraded. And similarly, when we do that, the AA expressing cells compared to control cells, they grow really poorly. So just having a little bit more RB due to a failure to ubiquitinate it almost completely crushes proliferation in these cells, which we think is super exciting and cool. We found that this is due to the repression of cell cycle gene expression, which is kind of what we expected since that's the major role of this protein. So here we're looking at a bunch of cell cycle genes. You can see that when we express the mutant, which represses proliferation and accumulates again to higher levels, it really suppresses proliferation. We also wanted to look at the proteins. To do this, we collaborated with a friend and colleague at UNC also named Jeremy Purvis. His lab has developed this technology called 4I, iterative indirect immunofluorescent imaging, which I spelled wrong, it looks like, or I truncated there, sorry. But they didn't develop this technique originally, but what they've done is they've taken this technique and they've applied it to the cell cycle. This is a technique that allows you to fix and stain cells, image them, and then strip them and restain them and re-image them. And they can do this for upwards of 40 or 50 proteins now. They have a whole panel of cell cycle proteins. What I'm showing you in the violin plots above are cell cycle proteins that correspond to some of those genes I showed you. You can see in the middle of this experiment, particularly here, in the AA expressing cells, there's much less of this one protein CDC6, also CD21 over here, compared to the controls. That's these sort of lopsided violin plots. Interestingly, by the end of the experiment, by 14 days, you can see it's sort of normalized. And actually, shortly thereafter, these cells start proliferating kind of normally again, suggesting that they're generating resistance. And we've looked, they're still expressing the mutants. So we don't know why they're coming out of this arrest or what's driving resistance. And this is something we're super interested in figuring out in the future. So this is sort of the canonical CDK4/6 RB pathway, which I introduced you to a few slides ago. We would now put Cyclin F in this pathway. We would, because it's we think it's ubiquitinating P130, similar to Cyclin D and CDK4/6. It's putting downward pressure on this pathway, inactivating not by phosphorylation, but inactivating due to protein degradation. To kind of further test that, we looked back into the DEP map, which has not only done loss of function CRISPR screens, but it also has screened about 500 of these cell lines with more than 1,000 different drugs in the idea of doing drug repurposing. If we say what drugs are most similar to Cyclin F loss, you can see palbociclib, this is a CDK4/6 inhibitor developed by Pfizer used clinically, is the #1 drug most highly correlated with Cyclin F loss. And that really suggests, again, like I showed on that previous slide, that Cyclin F is promoting proliferation, doing something through that pathway, we would argue, through degrading P130. Ribociclib, another 4/6 inhibitor, CDK4/6 inhibitor, is number 24 out of, again, more than 1,000 drugs. If we reverse that analysis too, we can say what genes look most like palbociclib, right? And again, you see Cyclin D, CDK6, and CDK4 at the top. And again, out of 17,600 genes, Cyclin F is really near the top. We think this provides really compelling evidence, probably more compelling evidence than all of our in vitro biochemistry and cell biology, that Cyclin F is really implicated in this pathway. Okay, so that's the end of the first part. The second part of my talk, that part was recently published. The second part of this talk is going to focus on deubiquitinases in cell cycle and how we identified enzymes and substrates there, again, using large data sets. This is totally unpublished. This is also sort of a work still in progress, and I'd welcome any feedback from anybody here on Zoom or otherwise. Okay, so this is the cartoon of the ubiquitin pathway I showed you maybe 20 minutes ago now. What I did not build into this pathway, but which JoAnn alluded to that we are super interested in, is the role of deubiquitinases. So deubiquitinases, or DUBs as they're conventionally called, are catalytic proteases that remove ubiquitin from substrates. And at least in terms of protein degradation, they can antagonize the degradation of protein. So when a DUB is increased, you can think of it as sort of activating its substrate by preventing its degradation. In humans, there's approximately 100 deubiquitinases. And while the role of ubiquitin in cell cycle is, I would say, unequivocal, it's not so clear. The role of deubiquitinases is much less clear. And it's not even clear which deubiquitinases are actually involved in cell cycle. We thought we could probably predict this by doing something that's sort of like really simple. And that is just saying what deubiquitinases actually change in the cell cycle based on transcriptomics. A lot of the E3s that change in the cell cycle, like Cyclin F, are themselves regulated at the level of their own transcription. There's a really beautiful data set, which was put together by Jim DiCaprio's lab at Dana-Farber, which has collected a whole bunch of transcriptome data sets that were performed throughout the cell cycle. We went into that. If you look at like the canonical cyclins, which I introduced you to earlier, you see that all of the studies in those data sets that are curated by that website, all of those studies find all of the cyclins. So, and things that are not cell cycle regulated obviously don't show up, or they maybe show up sporadically in one study or another. So we looked at all of the deubiquitinases. There's actually only 6 deubiquitinases that show up in 2 or more studies as being cell cycle regulated. I'm showing those to you here. 3 of them already have pretty established roles in cell cycle. So USP1 is involved in S phase progression. USP2 controls cyclin D actually, and USP37 is thought to antagonize ubiquitination by the anaphase promoting complex, which our colleague Cole Ferguson works on. We were interested then in these other 3 to see if maybe we could get some insights into how they might be involved in cell cycle. So we looked at these 3, and we needed a way to sort of further get an idea or prioritize them and get some maybe insight into what they might do. To do this, we turned to these large protein-protein interaction data sets that are available in the BioGrid. And if we look at the protein-protein interactions, or at least the high confidence protein-protein interactions for these 3 DUBs, 18, 53, and OTUD1, we noticed that for USP18, it interacts with a protein, or has been reported to interact with a protein called SCIP2. And this got us really excited because SCIP2 is also an F-box protein and is also already known to be involved in cell cycle. So SCIP2 is an F-box protein like Cyclin F, whose levels go up and down in the cell cycle. And its major role in cell cycle was first described by Michele Pagano's lab at NYU, where he showed that SCIP2 is involved in the degradation of cyclin-dependent kinase inhibitors, these proteins called P21 and P27. So P21 and P27 would normally inhibit cyclin CDK activity. So SCIP2 drives cell cycle by promoting the degradation of these inhibitors, allowing the cell cycle to proceed. We wanted to do something really simple and just validate the observations we saw in our computer screen. So we synchronize cells by taking away serum and release them into the cell cycle. And it looks like USP18 levels go up at the same time that SCIP2 levels go up. That's pretty cool. We thought, oh, maybe USP18 is deubiquitinating SCIP2. We could also recapitulate that interaction that we saw in these protein-protein interaction databases. So if we IP SCIP2, we pull down USP18 and vice versa. And that's this data over here. So we felt pretty good about this. We were like, okay, it looks like 18 is cycling, it's regulating SCIP2. This is pretty neat, right? So then we had to figure out what USP18 is. So it turns out USP18 is not a canonical deubiquitinase. In fact, some of the best work, or perhaps the best work that's been done on USP18 is done by your colleague here, Dongler Zhang. USP18 is actually involved in the interferon pathway and is actually involved in the ISGylation pathway. So ISGylation is regulated by a protein called ISG15. And ISG15 is actually a ubiquitin-like protein. So this is ubiquitin here. This is ISG15 on the bottom. It actually looks a lot like 2 ubiquitin molecules stuck together. And whereas this is sort of the canonical E1, E2, E3 ubiquitin pathway, there's a similar pathway for regulating the conjugation of ISG15 onto substrates, only unlike the ubiquitin pathway where you have a couple of E1s and a few dozen E2s and hundreds of E3s, there's actually just one of each of these in human cells. So it's a very simplified system that responds to interferon and drives the ISG-alation of substrates. You can see this when you treat cells with interferon like we've done here, and then you blot for just ISG-15. You see the formation of these giant conjugates of ISG-15 onto substrates, much in the same way ubiquitin would get conjugated onto substrates. You don't have to do that with anti-interferon either, though. What you can do is you can just express each of the different components in the pathway, UBA7, UBCH8, and HERC5. When you do that, you can drive ISG-alation like I'm showing here. And if you leave out the E1 or make mutants in these different proteins, you can totally kill that. So we're going to look at a bunch of graphs like this for the next 15 or 20 minutes. This suggests there's some ISG-alation. And then when you see nothing, that suggests there's no ISG-alation. Okay, so what is USP18? USP18 is actually thought to be a d-ISG-alase. And you can see that here. This is our data recapitulating much other data from other labs, like Dong Zhang's lab. Here we've induced ISG-alation. When we add wild type USP18, you can see it completely reverses ISG-alation. This is done in cells. And then if we express a catalytically dead mutant, you can see it doesn't do it. So this is clearly a d-ISG-alase. So we thought, well, maybe it's a d-ISG-alase, but also is a deubiquitinase. That would be kind of cool. And maybe that had been missed so far. So we did a really simple experiment. We transfect dubs into cells. We pulled them back out. We think this is like a nice way to do this because they maintain their appropriate folding and post-translational modifications. And we can monitor dub activity by just mixing them with diubiquitin substrates and watching the conversion of diubiquitin to monoubiquitin. And you can see here when we did this, we were super pumped. I was like ready to write a paper. I was like, look at this. We're getting deubiquitination using USP18. This is like totally awesome. USP18 has some activity towards ubiquitin. And everybody's missed this and we're like patting ourselves on the back. The one caveat of these experiments, though, is that you're pulling these out of like 293 T cells and you don't know what else is coming down. So my really smart postdoc was like, maybe we should use like a cysteine catalytically dead mutant just to make sure this is actually USP18. And we were really sad when we did that because it turns out that whatever is doing the deubiquitination in this assay has nothing to do with USP18 because when we make the C to A mutant, it looks exactly like the wild type. Okay, so whatever this activity is not USP18, whatever is doing the deubiquitination. So then we were like, well, okay, what is doing the doing the deubiquitination? We went back to those proteomics data sets that had looked at protein-protein interactions, and we noticed in addition to SCIP2 being on there, another interacting protein with USP18 is this other dub called USP41. We looked in another PPI data set, this one from Steve Geeky and Wade Harper's lab, this is called the Bioplex. In fact, USP41 is reported as like the only interactor with USP18. Okay, so now we're like, okay, now we need to figure out what USP41 is. Turns out USP18 looks exactly like USP41. So they have like 80% identity and similarity. So now we have two deubiquitinating or d-ISG-lating proteins that look like they're interacting with each other. Maybe one of them is a deubiquitinase, maybe one is a d-ISG-alase, and we think we need to figure this out. So we could recapitulate the binding between them by introducing them both into cells. My postdoc cloned USP41. There's like almost nothing known about it. In fact, I think he was probably the first person to clone this thing just a few years ago. We put them both into 293 cells, and we can show the interaction no matter which way we do the IP. And these things are pretty similar. So if you look at 18 and 41, through their USP domain, they're over 90% identical. In the N-terminus, they're about 50%, 40% identical, excuse me. And this is going to become really important. 18 has about 14 extra amino acids on its C-terminus that are lacking in USP41. I'm going to say that again. There's 14 amino acids at the very end of 18 that are not present in 41. This turns out to be really, really important. So these things look just like each other, particularly in the domain that does d-ISG-alation. So even though they look like each other, though, USP41 is not a d-ISG-alase. We have never been able to see any d-ISG-alating activity when we put in 41. So again, here's controls where you see all this ISG15 conjugation. We put in USP18 in that second lane, or where my marker is right now, completely de-ISGylates everything. We make the mutant in 18, doesn't do anything. 41 looks just like the mutant. We get no de-ISGylating activity from USP41. So now we're like, well, what could possibly be different between these things? This doesn't make a lot of sense. So we started comparing 18 to 41. We made some truncations to make 18 look a little more like 41. We take off its N terminus, we take off those last 14 amino acids and its C terminus. What I want to draw your attention to is that whereas USP18 is a great de-ISGylase, when we chop off those last 14 amino acids in the C terminus, so now it looks a little bit more like 41, now it doesn't do de-ISGylation at all. So now 18 looks like 41. It's a terrible de-ISGylase. So something in those last 14 amino acids, we don't really know for sure what, is really essential to do de-ISGylation. And we've actually been able to figure out where the specific amino acids in the USP domain that are required for this. We can actually turn 41 into a de-ISGylase just by making a few amino acid changes. I don't have time to take you through all of that. Okay, it turns out too that USP41 actually binds to skip2 even better than USP18. So I showed you when I started this that we kind of got into this because 18 could bind to this F box protein skip2. Turns out 41 binds much, much better. So if you look here, here's the binding of 18. You can barely see it because we haven't overexposed this, but here's the binding of 41 to skip2. And this works in both directions as well. So USP41, remember, it lacks these last 14 amino acids. It is a terrible de-ISGylase, but it binds really good to skip2, which is this F box protein that's involved in cell cycle. And if we knock it down, if you put 41 into cells, you can see it will do skip2 deubiquitination, whereas 18 does it much more poorly. So we think that probably 41 is what's actually deubiquitinating skip2. So it binds it better. It looks like it does deubiquitination, but not de-ISGylation, whereas 18 does ISGylation, but not deubiquitination. When we knock it down by siRNA, you can see we've used 4 different siRNAs here. All of them reduce the levels of skip2. It's these lanes versus this lane here. These are synchronized cells in late G1 phase or early S phase. And you can see when you do that, the CDK inhibitor P27 is now elevated. So what we think is happening is 14 is coming up. It's deubiquitinating skip2 to protect it. And this is allowing it to grade P27. Similarly, when we deplete USP41, we can reduce the ability of cells to enter the cell cycle. So here we are looking at EDU labeling. This is marking cells going into S phase. We first synchronize them and then release them. If we deplete USP41, you can see we see much fewer cells in S phase. And this is reflected in our quantification on the right. So this, we were looking at this and we were thinking, well, what do USP18 and 41 look like in other organisms? Because David Commander and colleagues had published a really nice review on this a few years ago. And in that, they noted that while humans have 18 and 41, actually most other organisms do not. So other organisms, like in this case, we're looking at fish is where I have my pointer, or even in mice, they only have one of these. So Dong Zhang has worked on this, this enzyme in mouse. It's called UBP43, just to make it confusing, I think. But mice actually even only have one of these. So humans are very unique in that they have 2 of these. We're wondering, well, if one is doing ubiquitin and one is doing ISG15 in humans, what could be the situation in other organisms? So we looked at these different, we cloned the other USP18/41 orthologs. When we look in humans again, you can see USP18 only does ISG15. So this is looking at what's called a charging assay. So these are suicide substrates that get conjugated to the dub when we mix them together in vitro. You can see we see really nice charging of USP18 with ISG15, but not with ubiquitin. Whereas we look in either fish or mice, you can see actually both of these can get charged with both. So in fact, the fish one is really good at getting charged with either ISG15 or ubiquitin. the mouse one too, it actually does ISG15 and ubiquitin as well, suggesting that perhaps in these other organisms outside of humans, maybe these things are sort of doing double duty. And this is really an attractive idea because a long time ago, people had knocked out ISG15 in mice, and they showed that, knocked out ISG15 in mice, and you can see, yeah, so the mice sorry, I want to make sure I say this right. So the mice are very sick if they are forming ISG15 chains or not forming ISG15 chains. And this could be, they thought this would be reversed by just getting rid of the deubiquitinase. But that's not the case. In fact, the mice that are lacking ISG15 and UBP43, you can see in the open circles here, are just as sick, suggesting that there's some other role for UBP43. I think I said that backwards. Let me say it one more time. The mice lacking UBP43, this is the mice version of USP14, die within a few weeks. The idea was that they're forming ISG15 chains that they cannot then take apart. But when you take away ISG15, that does not rescue the phenotype of these mice, suggesting that this enzyme is doing something else besides just doing de-ISGylation. So then we turn to the mouse version of this, which is called UBP43 or mouse USP18. We asked if maybe that's regulating the mouse version of SCIP2. So we can monitor really nicely SCIP2 ubiquitination in a test tube. I'm showing that here. But when we mixed it with UBP43, which is again mouse USP18, actually it didn't do anything. So we're sort of disappointed. We're like, well, maybe this works in mice or in humans, but not in mice. Maybe we don't know what's going on here. Maybe it's not a dub after all, at least in mice. But then we noticed something really, really interesting. We're looking at the exon structure of the mouse gene, and we noticed that actually those last 14 amino acids that I referred to earlier, they actually form their own exon in the mouse gene. And it looks like you can splice those out. So it looks like there's splice isoforms of the mouse version of USP18, of UBP43, that lack that C terminus. Remember, I told you earlier that C terminus is super important for determining whether or not this thing is a de-ISGylase or a deubiquitinase. So there's one lacking those last 14 amino acids. This one looks like 41, whereas that full length one looks like USP18 in humans. So that motivated us to then go back and look at a splice form that lacked those last 14 amino acids for the mice version as well. You can see here when we take away the C terminus of UBP43 or mouse USP18, that totally kills deubiquitination again, just like in the humans. And again, when we take away that C terminus, it binds to SCIP2 really, really, really good. And if you've been following along, you should know this looks a lot like the human story I told you earlier, right? So those last 14 amino acids, which encode their own exon, which can be spliced out, that makes the mouse version look just like 18 or 41. And this suggests the possibility that maybe what mice are doing is they're changing the splice isoform of UBP43 or mouse USP18 to make it look like the human 18 or 41. And then humans just duplicated this gene. So now we have one that does these two different activities. And consistent with that, if we now take UBP43, I told you it doesn't deubiquitinate SCIP2 at all. But if we use one lacking those last 14 amino acids, now it looks like a really good deubiquitinase. Whereas this one, the full length, is a really good de-ISGylase. Similarly, these are the UBP43 knockout MEFs, which were sent to us by Dong Zhang last year. You see when we knock out UBP43, SCIP2 levels go down. Presumably it's getting ubiquitinated and degraded, and its substrate P27 goes up. And we're in the process of seeing if we can rescue the proliferation defects in these MEFs by knocking out P27, which is again the CDK inhibitor. Okay, so what do we think is happening? To sort of summarize this, we think that in humans, you have two different genes, one called USP18, one called USP41. One is a de-ISGylase and one is a deubiquitinase. These two bind to each other, which is kind of interesting and cool. In mice, where we know these two things can dimerize as well, I didn't have time to show you that, 18 or 43 are basically just one gene, but you can splice off those last 14 amino acids, making one a dub and one a de-ISGylase. And that dub one binds to skip2. This is an F-box protein. And by doing so, it's going to prevent the degradation of skip2, allow it to degrade its substrate P27 to promote cell cycle. And we think this is like a really cool idea. You know, your colleague and my friend, Eric Bennett, published a paper a long time ago when he was a postdoc, maybe 15 years ago, where they did pull downs of upwards of 50 human deubiquitinases. One of the major things, the clients that they pulled out with all of these were E3s. And I think the way most people thought about it at the time, or the way we've at least thought about it, maybe naively, is that you have an E3 binding to a deubiquitinase so that you can sort of coordinately control the ubiquitination of its substrate. But that might not be true, actually. It could be the deubiquitinase is there to prevent the E3 from itself getting degraded. Many E3s get ubiquitinated in the process of doing ubiquitination, but that's not a really good thing if you want to keep that E3 ubiquitin ligase around, because if it gets ubiquitinated, it's going to get degraded. So maybe one of the major important reasons that DUBs sit on E3s is not to antagonize the ubiquitination of substrates, not that that doesn't happen, but they're there to protect the E3s themselves. And we think that's what's going on here. Okay, so that's it. I want to thank everybody for their time. So all of this work was done by, really driven by two people. One is Taylor Enrico, and the second is Thomas Bonacci. But I don't know, we are again, a small group, and we've had lots of different contributors to this work. We've had great collaborations with people both at UNC and elsewhere. So it would be remiss not to mention Nick Brown and Jeremy Purvis's lab at UNC, and Seth Rubin at UC Santa Cruz. Of course, I would like to thank the NIH and GM for funding, the American Cancer Society for funding for some of our more cancer-specific work, and then the University of North Carolina's Cancer Research Fund for other funding. So, and thank you for your time, and thank you for having me out, JoAnn.
Wonderful, thanks. So I'm going to start with the first question. So how does that 14 amino acid C-terminal peptide region affect the specificity, the protease specificity? Because I'm assuming it's still being proteolized through the USP domain, which is the catalytic cleft for the substrates, right?
Yeah, that's a super great question. And one that I frankly don't have like a very good answer to. So we don't know why those 14 amino acids are important. We don't know where they are. We've looked in things like AlphaFold and stuff like that. It looks like it's sort of unstructured and probably hanging off of the USP domain. And we don't know what it would be doing to allow the DUB to do deisgylation. So really what it does is we think it allows it to switch between these two things, or at least it is required for 18 to be a deisgylase. But the reason for that is like not known. We're trying to like sort of take like a molecular approach, which is to sort of step back through that thing and see like where it becomes essential, and also to mutate the different amino acids. And then we're starting to think about doing some more structural work to get an even better idea. Again, the people that have done structures of these things have actually, like most structural biologists, they trim them down to like things that all fold nicely and stuff like that. And this has been invisible in the structures so far of USP18. So we don't have like a good explanation, but I think this is like the exact right question to try to figure out.
Great. Okay, so I'll take questions from the audience first.
Hi, I wanted to ask what you think the purpose of evolving these two separate like USP18 and 41 are in humans specifically, when you've seen that it like, I guess, lower eukaryotes that there's only one enzyme performing both of those functions.
Yeah, that's an interesting question too. So it's very clear that the reason you have two in humans is due to gene duplication. In fact, they're really pretty close to each other on the same chromosome. I don't know if there's necessarily a great reason to have two if you can do it with splicing. It could just be that you had a genome duplication that then became fixed, and that maybe mice have the better way of doing it. I don't know. I don't want to say like one is necessarily like better or worse than the other, but I think that they can be potentially redundant. So 41, I will say, binds to skip2 really, really well, probably the best of all of these. So maybe perhaps the deubiquitination is a little bit more of skip2 is really important in human cells versus in other systems. Whereas you could sort of, you could get by with having them be constructed into the same enzyme and just doing it through splicing. It allows you more like dynamic control of the system too, because you don't have to, you can make one protein or the other. You don't have to worry about having the splicing. We don't know at all how the splicing would be regulated at this point, developmentally or otherwise.
Hi, Michael. I learned a lot. Thank you so much.
Thanks.
I was wondering when you mentioned that one of your hypotheses is basically that little tail could be inhibiting the ubiquitinases to damage themselves, right? So it would be like the same idea would be like they will not be ubiquitinases because they have that little tail or because they don't have that. That's my first question.
Mm-hmm.
And because thinking as venoms, that happens a lot.
Okay.
And have you ever tried to use just that part, those 14 amino acids, and see if they inhibit the activity that you're studying?
So I'll tell you what we have. So when we think those 14 amino acids are important for deisgelating activity, but probably restrictive for deubiquitinating activity. What we have done is we've taken that tail and moved it onto 41. That's not sufficient to make it a deisgelase, but there's only, I think I showed this, there's within the USP domain, they're like almost 95% identical. There's actually only a handful of amino acids that are different. So if we move over the 14 amino acids, that will not make 41 a deisgelase, but then we only need to make one other amino acid change, and then we can convert 41 into a deisgelase. And this was sort of like something we did on the side because these assays are really robust. So it took a little bit of playing with, but we ended up comparing 18 to 41 to the mouse one, which we know can do both. And we can like convert it. I don't know if that gets at your question though.
All right, we're gonna move on to the next question. So Sylvia Gutkin has a question, and then we'll move to Jing Yang.
Perfect. So, Mike, so amazing presentation, and I really enjoy how well you explained it, even for me. So a quick question. So regarding the possibility of targeting in cancer, so if this is correct, so the possibility to block the associated deubiquitinase rather than the E3 ligase. So if that's the case, are there deubiquitinases that are, for example, associated with MDM2 or MDM4 that can be targeted?
I think, so maybe correct me if I'm wrong. I want to make sure I'm answering the right question. You're asking about, could we target the deubiquitinases therapeutically?
Yeah.
I think that's like an awesome idea. I love that idea. So I think, I don't want this to sound too rehearsed, but I've written this into many grants. So deubiquitinases, I think, are the most druggable, reasonably druggable target in the ubiquitin pathway. They have a catalytic protease that can be inactivated with small molecules. In the last, I think for a long time, all of the DUB inhibitors out there were probably pretty crappy because they were non-specific. But in the last 5 years, we've seen an absolute explosion in this space. So Genentech and Forma Therapeutics, and also Sarah Berlaghi's lab at Dana-Farber, have developed exquisitely selective and highly potent inhibitors to USP7. USP14 inhibitors that are highly selective have been developed. USP1 inhibitors have been developed, and they just entered from KSQ Therapeutics in Boston. They just entered clinical trials and talked about this. I think this past April. So I think that these are an awesome idea for drug targets, and they hold this amazing possibility that, you know, if you want to get rid of some a cancer protein that is non-catalytic or is some sort of a scaffold or something like that, that would be difficult to target directly, you could inhibit its deubiquitinase and then trigger its degradation just by harnessing sort of the innate degradative machinery to promote its destruction. So in this case, you can inhibit 41 in principle. This would allow for the deubiquitination or the ubiquitination of SCIP2 and the stabilization of P27, this really potent tumor suppressor and proliferative inhibitor that binds to the CDKs. And I think this is probably one of many deubiquitinases that would be worth considering as a therapeutic target in the future. So we've worked on other ones. I think there are many others out there. They need to have sort of the right expression profile in the tumors. Of course, the right sort of like collateral vulnerabilities in terms of what else is going to change. I think we need to think a lot about that. I think there'll probably be tumor specific ones like there are with other therapeutics. But I think this is a really nice idea. For example, I mentioned in the beginning, one of the other cell cycle deubiquitinases is USP2. This deubiquitinates cyclin D. So we already have drugs hitting this pathway. I mentioned them in the beginning, palbociclib and ribociclib. Perhaps bringing down cyclin D levels would be a really effective strategy in some of these cancers that are resistant to the 4/6 inhibitors.
Yeah, great question. Okay, I'm going to let you guys continue discussion at your individual meeting. I'm going to move on to Jing Yang.
Yeah, hi, Michael. This is such an interesting talk. So my question is this USP41 splicing or activities regulated by cell cycle checkpoint? Or I guess the more general question is really how the cell decide on which protein is going to be deubiquitinin at certain time. Is the target that is modified or the deubiquitinase is regulated?
Yeah, so that's a really good question. We don't know what, so USP41 itself is cell cycle regulated. I showed in, I can't remember which cells I showed it in, maybe T98Gs, that it comes up as they come out of quiescence. We see this in other cell lines. We don't know we actually know very little about like what's doing the transcription of USP41, if it's getting degraded, what's doing the degrading of USP41. In mouse cells, we have no idea what would be doing the splicing. So in humans, you just have to express one or the other, 18 or 41. So you can turn on 41 by itself. In mice, presumably you have to control the transcription, but coordinately control the splicing so that you don't have that C terminus on there. And we have zero clue what would be responsible for that, but that's a super interesting question that we hope to figure out.
All right, great. And I'll let you guys continue the discussion when you meet later today. Okay, so thank you, Michael. Wonderful talk. And thank you everyone for attending the presentation today.